be giving a broad overview of the basics of clinical trial design. I should start by saying a bit about who I am. I'm a professor of biostatistics in Newcastle University, and I'm interested in statistics, but specifically how statistics can be used with clinical trials. And I do research on how to make clinical trials more efficient with more innovative statistical approaches, but I also take part in a lot of clinical trials as a statistician. So if you're interested in finding a bit more about our group, we have a website here, and always very happy to be contacted if anything I say is of interest. So today I'm going to talk for about 15 minutes, and this is the outline of what I'll talk about. So I could really talk for probably 2 days about how to do clinical trials. I'm not going to do that, but just to say that this is a very high level overview and there's a lot more to it. And I think that's why it's great to have RDS and the Sir Bobby Robson team available to provide support when you're thinking of doing a clinical trial yourself. So I'll start off with talking about why clinical trials. I'll be focusing on oncology trials, but a lot of what I say is more generalizable to different types of trials. I know not everyone here today is working in oncology. I'll talk about efficacy and effectiveness and the difference between them. And then I'll talk about a way of thinking about summarizing the design of a trial, which is called PICO. And a bit of statistics, but hopefully not too much about how big a trial needs to be. So something about sample size calculation. And I'll finish off with pointing you to some further guidance and resources for planning studies. So we're interested generally in ensuring that treatments and interventions are safe and effective. And one way to do this is with an observational study where we have a look at patients who receive an intervention in practice against those who don't and compare outcomes. But this can really give us misleading answers. So a really nice example of this is in Dickerman et al. And this is looking in the context of taking statins and its effect on cancer risk. So several observational studies had looked back retrospectively at individuals who received several years of statins and compared their outcomes in terms of whether they developed cancer or not, comparing those who got statins with those who didn't. And a lot of these observational studies found huge protective effects of taking statins and were published in very high profile clinical journals. This is an example of one of the analyses where you can see that they compared those who used statins and they looked at people who had developed cancer and people who hadn't, and they showed that it was much less common amongst cancer patients to have taken statins compared to not taking statins. So this appears to imply that statins are a really good thing to take to prevent cancer. However, these studies turned out to be very misleading, unfortunately, and the answer was that statins don't actually provide any protective effect against cancer. And the main issue here was the way that these observational studies were being done. They were looking back in time at individuals who had 5 years or something like that of taking statins and comparing them against those who hadn't had 5 years of taking statins and in fact never taken statins. And the idea here is that we're comparing two groups that are not really comparable, because people who die from cancer are less likely to have lived long enough to take 5 years of statin compared to those who hadn't died from cancer. So we're artificially introducing a bias here by doing it in this retrospective observational way. So patients who died from cancer, patients who had taken 5 years of statin appeared to not have cancer so often. And this is really due to a bias that we can avoid to some extent by doing observational trials in a better way. But even if we do observational trials in a perfect way, we still have biases that can occur due to various factors. So a better, higher quality way of testing a new intervention versus an existing intervention is a randomized controlled trial. And this is what we would call the gold standard study design in terms of giving the highest level of evidence about the safety and effectiveness of a new intervention. And the idea here is that we're randomizing patients. So we're seeing patients who are eligible for the trial. We're making sure they're happy to be recruited to the trial. And then we're randomizing them between the control arm, which might be their current standard treatment, and an experimental arm, which is the new treatment that we're seeking to compare. And in each group, we're following up the patients and looking at their outcomes. I will say more about all of this later on, but just as a high-level overview. And then we're comparing the outcomes between the two groups and seeing whether there is a difference between the two groups. And if there is, we can say that it's due to the new treatment because we've randomized patients. There are much less opportunities for biases, like I've given an example of before. So just to compare observational studies and randomized trials, observational studies are subject to what we call confounding bias. So if we have two groups that we're comparing, and there's a third factor which influences both which group a patient is in and their outcome, then we can get confounding, which appears to lead to differences between the two groups when there actually aren't any due to the treatment that we're testing. Whereas randomized trials are not subject to confounding, and we can exclude confounding as a reason to see difference between the two groups. Observational studies can be subject to selection bias as well. So if we're controlling the groups that we're comparing in some way, then we might be maybe unethical and put patients who we think will do better in the group that we're wanting to show is better, for example. Whereas in a randomized trial, we can't do that, and therefore they're not subject to selection bias. On the other hand, observational studies are generally a lot cheaper to do, whereas trials can be very expensive. So I'm not saying there's not a role for observational studies. They are the appropriate thing to do. And in many cases, we can't actually ethically randomize patients between two interventions. So for example, if we're looking at whether smoking is associated with lung cancer, we can't ethically randomize patients to smoke or not. We have to look at observational data in that case. So I've talked about randomized controlled trials, but in fact, trials are over several phases, especially for drug trials. Drug development is highly regulated by international bodies, and it consists of several phases of clinical trials. So it starts at phase 1, which is the first point at which a drug would be tested in humans. And a phase 1 trial would be to demonstrate the safety and make sure it's not unsafe, and to find a dose that is sufficiently safe to justify testing in the next phase, phase 2, which is looking generally at shorter term efficacy in a moderate number of patients. So this might be looking at fairly short term outcomes of patients, and it might be in quite a highly selected group of patients, but often still will involve randomizing, not always in oncology phase 2 trials. So, the purpose of phase 2 trial is to demonstrate that it's worthwhile doing a much larger and longer term, more expensive phase 3 trial, which is looking at longer term effectiveness and safety on a generally a much larger number of patients and can take several years to do. So that's for drugs. For non-drug trials, so surgery is a good example. There's generally less of a distinction between phases, but there is more for both drug and non-drug trials, a distinction between trials focusing on efficacy, which was the phase 2 objective in the previous slide, and effectiveness, which was more the phase 3 objective. So these two concepts are very highly related. Efficacy is, does the intervention work, maybe under ideal circumstances, and effectiveness is more, does it work in real life settings? So does it work in the NHS, for example? And generally you can't get effectiveness without efficacy, but you might have efficacy without leading to effectiveness if, say, the drug is efficacious and it works if the patients take it, but it's so much of a burden to take that it's not actually effective because no patients take it in practice. If we're looking at trials that focus on efficacy versus effectiveness, generally the effectiveness trials will be more likely to have a wider enrolment criteria for patients. They're more likely to be multi-centre trials involving maybe across the UK or even international sites with a heterogeneous population recruited. They're more likely to be larger, and they're more likely to have longer term follow-up of patients. So now we've considered the different phases and the efficacy and effectiveness, we can consider the design of the trial. And this is nicely summarized in an acronym called PICO. So P is for participants, and this is which participants, which patients will be recruited into the study and at which point in their patient journey. So soon after diagnosis, or maybe a long time after diagnosis, after they've had a standard treatment, for example. And we have to also consider how will they be recruited? So how are they going to be approached and told about the trial?
Yeah, Mr. Norbin. Yeah. Did you find him at all? No.
I'm sorry, there's someone who's muted. Thank you. I think that's muted now. The intervention is the I, and this is basically what is the intervention and how will it be delivered in practice. So that might be, if we're looking at a drug, what's the dosage and how is it administered? If we're looking at surgery, then what does the surgery involve and how are we going to train surgeons to do it if they're not familiar with it already? The comparator is the control treatment, and in drug trials we often have a placebo that is used to ensure that patients and researchers don't know which arm they've been allocated to, but placebos are not always used, and sometimes it's not possible to use them, such as a surgical trial. Although there is a concept of sham surgery, which is an interesting debate. Outcomes, which is the O. This is how might the intervention we're testing improve patient health, and therefore how should we track whether the intervention provides a benefit. So how are we going to measure efficacy or effectiveness? But also, how often will we assess patients, and how burdensome will be doing this be, and might this put off participants if they have to fill out a battery of questionnaires every month? That takes hours to do. So generally, we're going to now move on to choosing a sample size. Generally, we can think of a clinical trial as an example of an experiment where we're testing a hypothesis, and we're following ideas from hypothesis testing. So this involves specifying a null hypothesis. Normally, this is the hypothesis that is the default, and we want to gather evidence about in order to potentially reject if it's not true. So generally, the null hypothesis would be the more conservative assumption that the new intervention arm is worse or equal to the comparator arm in terms of the primary outcome we've chosen. And we are doing the trial with the objective of rejecting this null hypothesis if it's not true. Then we're going to gather data from the study, so the outcomes in each arm, and we're going to summarize this data into what we call a test statistic. And the idea is this test statistic is some value, it's a number, and that number will be random, but it will have a distribution that we know when the null hypothesis is true, so that we can compare its actual value from the data in the trial, and we can see whether the test statistic is consistent with the null hypothesis or not. And if it's very different to what the distribution should be under the null hypothesis, we can reject the null hypothesis. We've gathered evidence to say it's not true. So what does the sample size depend on here? So it depends on the significance level, which is the chance of incorrectly rejecting the null hypothesis, or in this case, incorrectly recommending the experimental treatment when it's not actually better. It depends on the target effect size. So what difference in the primary outcome are we hoping that there's a high power to find in the study? So what is the difference between the two arms that we'd like to have a good chance to find if it exists? The power itself is the chance or the probability of rejecting the null hypothesis when the true difference is actually equal to the target effect size. And so normally that would be a high value like 80 or 90%. And it also depends on some other things depending on the scenario, usually to do with the distribution of the primary outcome in the control arm. So let's have a look at an example, just bringing together the PICO and the sample size calculation. So let's imagine that we want to randomize patients with metastatic lung cancer that have very poor prognosis. So those would be our participants, and we're going to randomize them between a new drug that we're testing, which is the intervention, and the existing best available treatment, which is the comparator. So it's thought that the new drug may hopefully improve or decrease the 5-year mortality rate. So that's the outcome that we're going to use to power the study. The null hypothesis then is that the 5-year mortality rate for patients who have the intervention is equal to or higher or worse than the comparator. Just to remind you, that's the null hypothesis that we want to reject. We want to show that the new treatment is better than the comparator. The data collected is the 5-year outcome from patients who have been recruited in each group. And the test statistic will be some way of summarizing that data into a number that we can compare and decide whether to reject the null hypothesis. So if we wanted a 5% significance level, so a low chance of falsely saying the new treatment is better when it's not, and 80% power, so a high chance of showing the treatment's better when it actually is better, and we assume maybe that the 95% mortality rate in the control arm, depending on that would obviously depend on which participants we're recruiting and evidence that's in the literature. And then we might be hoping that the new drug would provide a 10% improvement, so a reduction to 85% mortality rate in the experimental arm. Then putting all of this into an online calculator, for example, and this is a fairly simple case where you can do this, that putting those values in leads to a sample size required of 282 in total. We might want to consider that some patients won't stay in the trial for the whole 5 years, and they might drop out for other reasons. And if that's expected to happen, then we might want to increase that so that we have around this number at 5 years. So that's just to give you an example of a sample size calculation and a design. But of course, it's really going to depend on your case, and it's all very depending on the trial that you're wanting to do. So it's very difficult to give a broad overview that covers every possible circumstance. And for that reason, I would definitely recommend that you talk to suitably qualified clinical trials methodologists. So hopefully through the RDS, and if you're doing a cancer trial, through the Sir Bobby Robson team. And do this well in advance of a funding deadline, because it takes a good number of months to put together a grant application that has a high chance of being funded. I think Rebecca's going to talk more about that later on. If you're interested in some further reading, my research interest is innovative trial designs. So one example is adaptive trials, which allow you to make changes to the trial as you collect data. If you're interested in that type of thing, then this paper in BMC Medicine provides a hopefully accessible overview of adaptive designs. And people in my team are very keen to collaborate on implementing these in practice. So if this is of interest, please let us know. If you're interested more in phase 1, then there's a paper by Wheeler et al. In BMC Medical Research Methodology. And I didn't talk much about biomarker guided trials, but this is an emerging area of trial design that's also of interest to our group. And there's a paper in PLOS One that might be of interest there. And I think I've said all of this already, but a bit about our group, the Biostatistics Research Group. Well, we have a wide variety of expertise in both methodological development, but also application in real studies. And we work very closely with the clinical trials unit to deliver statistical support in real studies. So please do have a look at our website and get in touch if it's of interest.